Hey guys, so we're going to continue our conversation about biochemistry by now going over chapter 7.1, which is all about the genetic code. Okay, so first we're going to go over the different types of RNAs that you have. So you have messenger RNAs, which carries information from the DNA to your ribosome. We have tRNAs. This means transfer. tRNAs transfer amino acids using a anticodon. Okay, so you could think transfer move. This is the easiest one to remember here is ribosomal rRNAs, just means ribosomal RNAs, and these are just the structure and different enzymatic parts of your ribosome. So, what are codons? So, codons are triple coded, meaning that we need three nucleotides to make one amino acid. Okay. So, we need to go over a few important codons. So, AUG is your start codon, which codes for methionine. Okay, and we have these are very important for the MCAT that we see a lot. You need to make sure that you know your stop codons. Okay, we should all know start codons by now, just because we've been through some school. But if you don't know your stop codons, that's fine. I didn't know them at first, but you need to know those are UAA, UGA, UAG. To me, these are very easy to remember. Right, we just always have a slight switch in the second codon. I mean, in the second base. So codon is degenerate, meaning that multiple codes can make different amino acids, except for methionine and tryptophan. Okay, those are not degenerate. And wobble means that your third base is variable, meaning that that's the part that lets you know how many different codons that it binds to. Fun fact, except in your stop codons. So in your stop codons, it's the second base that changes the most times. So yeah, I mean here you have the one at the end that changes, but usually you would see the third base. So the third base we call it the wobble base, and we call it the wobble base because we can move it, right? You can change the wobble base, and then it wouldn't change your protein. So next we're going to go over mutation. So silent mutation, very easy, no amino acid change. Minces mutation, one amino acid is substituted. So one big one is sickle cell anemia, where your glutamine is substituted for valine. Okay, that is a Minces mutation. Nonsense mutation means that you have a premature stop codon, and then frame shift. When you add not multiples of three, you're going to move your codon down and you're going to change the strand of the DNA. Remember, we know that three codons make one amino acid. But if you add one codon to, if we add one base into a codon, right, into a strand of like DNA or whatever, we add one base into there, it's not a pair of three. So now you're actually going to change it. Remember, only pairs of three don't change it. If you add a pair of two, you've caused a frame shift mutation in the DNA. Okay, so next we're going to go over chapter 7.2, which is all about transcription. So, see you there.